This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 2 for November 2009. Hello, everyone. I'm Vincent Racaniello, and with me today is Dick Depomier.
Hey, Vince.
How are you?
Well, thank you. How are you?
I'm well. I'm having a busy day, but
yeah, we all
fine.
are.
It's good to be busy.
It is.
Did you lecture in your parasitology course today?
No, I lectured in my parasitic disease course. What's the difference,
What's
you
the
may
difference
ask?
between parasitology? Here, let me put it this way.
Please.
What's the difference between parasitology, parasitism, and parasitic diseases?
Okay. Those are 3 different terms. Let's begin with parasitology. The ology part means the study of.
Okay.
And the parasites imply a certain group, as we described in episode 1, namely the eukaryotic parasites. So it's the study of those parasites that parasitology addresses.
Studied at any level, molecular biology,
Correct.
disease,
Taxonomy, epidemiology,
systematics,
all of that.
All right, that's parasitology.
That's a parasitology.
And it's not what we called this podcast.
No, it isn't. It isn't, because we didn't want to limit ourselves to that concept, because what we're trying to do is stretch This Week in Virology and This Week in Parasitism into a common theme of parasitism itself. So now that's the second term you raised.
Parasitism.
Parasitism, that's why we call it this. Parasitism is the biological phenomenon in which one organism takes metabolic advantage of another organism.
All right.
That's
property of
parasitism. So viruses fall into this category.
Right.
Bacteria that are parasitic, that cause disease.
Are we going to discuss in this podcast parasites that are bacteria?
No, we're not.
We aren't.
We are not.
Why is that?
Because I'm not an expert in that area.
So we should have called this podcast Eukaryotic Parasitism.
Or This Week in Parasitism, parens eukaryotes.
No, that's too complicated. Are you open to considering bacterial parasites?
Of course.
We could get someone who knows about them.
Absolutely.
Do you know anyone who studies them?
I certainly do.
So maybe we could have them Skype in at some
Howard
point.
Schumann, in our own department, studies them.
Oh, this is a fabulous idea. Legionella pneumophila, for example.
Of course.
Okay. So I think we could have Howard on. I'm sure he would be happy to come up here and join us. He's
Precisely.
a great speaker.
He is. Alice Prince is another person who's an expert on bacterial parasitism.
What does she work on?
She works on drug resistance and transposons, transposable elements.
I know Alice. I'm having a meeting with her today.
Give her my best.
So we'll keep the bacterial parasites for later. So we're going to focus for the near future on eukaryotic parasites. That is organisms with a nucleus.
That's right. This is a big topic.
A defined nucleus separated by a membrane from the rest of the cell.
This is exactly correct.
All right.
So to get things in and out of the nucleus requires signaling peptides, etc.
Right.
And specialized pores, that sort of thing.
Now, your course is named Parasitic Diseases.
It is.
What does that mean?
That's the third term we mentioned.
Mm-hmm.
So what we're going to do here is to expose second-year medical students to the diseases that certain eukaryotic parasites cause. Now, I can't cover the entire field of eukaryotic parasites in this one course. There isn't time.
No.
I get 8 sessions with the medical students to cover what we consider to be the essentials of parasitic diseases. So in that course, we offer exposure to some worms and some roundworms first, and then some flatworms, namely the tapeworms next. And then we do the schistosomes, which are part of the trematodes. And then we shift gears and do the protozoans.
Are all these parasites that these students, when they're physicians one day, might encounter in
That's
their
how we try to couch this course in such a way as it allows them exposure to things that they will later on see in the clinics.
So these are not just exotic diseases.
They're not exotic diseases, and in fact, we see a great number of them being diagnosed in the diagnostic laboratory and also presenting themselves, let's say, in neurology, for instance, with regards to neurocysticercosis, which we will discuss at some time.
So the course is called
Parasitic Diseases. And in fact, the name of this book is called Parasitic Diseases.
How many lectures entails the whole course?
8.
Really? You just do the whole course is 8 lectures? Just you? It's for a few other people.
Well, it's not just 8 lectures, though. It's 8 lectures plus 8 breakout sessions.
Okay.
So we have an hour's worth of didactic material to present.
Which means you stand up and talk to them.
Yeah. Pretty standard. But then we break out into 3 large groups. And each one of us, there are 3 of us in our group that take part in this course. We sit down with them and we go through cases. And we discuss the nuance of their life cycles as to how they might involve the environment. And then what we try to do is emphasize the role that the physician can play in advising the patient and their family as to how to avoid future infections with these infections. So in order to do that, they have to know something about the life cycle.
Got it. Now, I understand you've won many teaching awards for this course. Yes, you have.
The answer is yes, but anybody who's taught this course has won those awards because this is a very interesting subject.
So the kids love the course,
They
right?
do. They do.
How many years have you been teaching it?
Since 1971.
So this is why Dick is the expert for TWiP. And that's why we can do this, because I couldn't do this show on my own.
Yes, you could.
And I think after you retire, you should continue to do this.
I have retired, by the way.
Yeah, well, I thought it was December.
No. It's last January I've been retired.
You're Professor Emeritus.
I am.
Well, you come to work every day.
I do. That doesn't mean that you stop working just because you've retired.
You don't stop thinking, right?
The moment you do, that's when your metabolic rate goes to 0.
So you have taught a very interesting and successful course for many years, and now we're going to translate some of that to this podcast, which is a great format for teaching, as you know, because we can reach a lot of people.
We can.
And the people we reach are interested. I don't know if all the medical students are interested in what they're taught.
It depends on how they're taught. I don't think it's a matter of what they're taught. It's a matter of how they're taught.
You think any subject can be made interesting if it's taught properly?
Sure.
All right.
Absolutely.
So, Dick, what are we going to learn today?
Today we're going to categorize parasites according to how they're transmitted.
Does that sound boring?
And the next thing, it could.
But you're going to make it interesting.
Oh, I'll try my best. And then the next thing we're going to do is to categorize the protozoans. We'll start with basic concepts of
general parasite.
Life cycle transmissions. Right. So let's begin then by saying that there are basically 2 major ways in which you can become infected with eukaryotic parasites. Okay?
Mm-hmm.
One is the direct method. And you'll never guess what the other one is.
the indirect method, I'll bet.
Well, actually, I would classify them differently. I would say you could either have a vector-borne infection or a non-vector-borne infection.
So we're changing it now to vector or non-vector.
That's right.
So a vector is some kind of an insect or arachnid.
Go on.
Or an animal, a mammal.
Could be.
Like a camel.
Well, that's a stretch.
So that's a vector. Anything that carries the parasite to you.
That's true.
For viruses, we typically think of ticks and mosquitoes.
Most of them are arthropods.
Arthropods.
That's right.
But for parasites, we have more than mosquitoes and ticks.
Oh, sure.
Name one, for example.
Black fly.
Sandfly?
Sandfly.
Excellent. All right, and then they're non-vector, which you're standing in dirt that has hookworms in it. I learned something from Radio Lab.
That's right, you did.
And they crawl up into the bottom of your feet. Isn't there another one where the people harvesting the rice, it goes into the schistosoma mansoni?
Look, listen to them. Come on, Vince, you could do this course.
No. Some vague recollections.
No, good recollections.
But malaria is transmitted by a vector.
It is.
A mosquito.
That's true.
And the name of that is?
Well, the Anopheline group of mosquitoes.
Group. So we're going to classify the parasites into vector and non-vector transmission, right?
And the reason why we're doing this is because this transcends protozoans and helminths. Okay, so you can't just say, well, for protozoans, there are 2 kinds of infections, vector-borne and non-vector-borne. Well, then you can go for the helminths, there are 2 kinds of infections, vector-borne and non-vector-borne. So just make vector-borne and non-vector-borne infections, and there you've got it.
All right.
Now,
That's good.
are there viruses that are vector-borne?
Yes,
But
absolutely.
of course there are.
West Nile virus,
Here's a very interesting question
yellow
now.
fever.
Are there any bacterial infections that are vector-borne?
Let's think. Let's let the clock tick.
So how would you classify rickettsia?
Rickettsia is one of those bacteria that need to grow inside of a cell, right? A eukaryotic cell.
Right. And how do you catch rickettsial infections? The bite of a tick.
Is that right?
That is correct.
So the
Or
rickettsia
a mite. A tick or a
are
mite.
free living in the tick, or are they within bacteria?
They don't. They parasitize the tick as well as their warm-blooded host.
So when the tick transmits it, is it transmitted as a free rickettsia or within a cell?
No, it's free. It's a free organism.
But these are bacterial parasites.
If you consider rickettsia as bacteria, yes.
Well, I think they are classified as bacteria.
I do too. I think they're prokaryotes. They're a little bit lower than regular bacteria because they can do less than a regular bacteria.
All right.
It's hard to grow them outside their host.
Did you ask if bacteria were transmitted by vectors? And the answer is yes, the vector being the eukaryotic cell. Is that a vector? Tick. No, sorry, you said tick.
Ticks.
Any other,
mites. Ticks and mites.
Any other examples?
Well, then you've got fungi to deal with. Fungi can cause diseases too, right?
Yes.
There are pathogenic fungi, lots of them. Are any of those vector-borne? Single fungus. I don't know one.
No.
I don't know a single fungus that has a vector.
Are they considered parasites?
If you get coccidioidomycosis, it certainly is. If you get basidio, no, that's not the right word. Well, there are lots of dermatophytes that cause disease.
Sorry to
Histoplasmosis is a fungal disease that gets into your lungs.
Is it considered a parasitic disease?
It causes disease. It's inside your lungs, of course.
So pneumococcal pneumonia is a parasitic disease?
If it's deriving metabolic aid from your cells, you bet.
So we have a very broad definition of parasites.
Of course we do. Of course we do. Parasitism.
Parasitism.
This is the act of one organism.
I'm beginning to get it, Dick.
Taking metabolic advantage of another organism. So you can move from viruses all the way up through helminths.
Okay.
And that's the point. So parasitology has classically been relegated to its own little niche, okay, of the eukaryotic protozoans and the eukaryotic helminths. Well, there are no such things as non-eukaryotic helminths, or for that matter, eukaryotic protozoans. So that if you were to characterize their mechanisms for transmission, then you've got just vectors and non-vectors.
So can we give some examples of...
We can, of course.
Is that where you were going next? I don't mean to drive this.
No, that's fine. So I can start with the protozoan parasites first. And we have, of course, two groups, direct transmission and vector-borne transmission. Okay, so I'll give you an example of...
vector and non-vector.
Yeah, that's right. I'll give you an example of a non-vector-borne protozoan infection. That's Giardia lamblia.
Giardia?
Giardia lamblia. That's the enemy of every camper.
It's in fresh water.
It's in fresh water. It's also infectious for other animals, beavers in particular.
Is it in the U.S.?
Oh, it's all over the place. It's
everywhere.
Everywhere.
When I was a
ubiquitous
PhD student, we would always hear of people going abroad and coming back with Giardia.
That's right.
Right. But then later we found that it was here
it's
as
everywhere.
well. So if you go camping and drink the stream water, which is cool and seems pure.
And it probably is, except for the fact that it's got some beaver feces in it. And the beaver feces has in it the cysts of Giardia lamblia.
Is that where the Giardia normally live, in beavers?
That is not normally. They normally infect a variety of hosts, and beaver is one of those hosts. We are another one.
So that's the main source of Giardia in fresh water?
It is. In fact,
cool.
It is.
So you take a little drink, and then in a few days or a week or so, you get intestinal issues?
Start to develop steatorrhea.
Steatorrhea.
See? This is the medical part of this course.
I love the term. What's the difference between steatorrhea and diarrhea?
Plenty. The organism Giardia covers over the cells in the small intestine and prevents the absorption of fats. So the fats continue on down until they get to the large intestine.
Fatty diarrhea, basically.
Then they're degraded by anaerobes. And anaerobes produce all these organic molecules as the result of their metabolism.
Explosive diarrhea? Smelly diarrhea.
Oh, you wouldn't believe. And abundant. And because you're not absorbing fats, there's also some weight loss associated with Giardia.
And you're dehydrating as well.
And you're dehydrating.
So what's the treatment for
Giardia?
Giardiasis, right?
Giardiasis.
Giardiasis.
There's a drug. There's a specific drug, and in fact, it was named after the group of protozoans that Giardia is part of. Can you guess the name of this drug?
No.
See, that's a car talk kind of quiz here, because it affects flagellates.
Flagellin?
Oh,
wait a minute, flagell.
Flagell!
Sorry.
Exactly. So the drug of choice for giardiasis is flagell. There's another name for this drug, of course. We don't use those names because that's a generic name. Or it's it's a proprietary name. The generic name for flagell is metronidazole.
Cool.
And metronidazole interferes with anaerobic metabolism.
And it kills the Giardia.
It does.
Are they anaerobic or are they facultative?
No. They're anaerobic.
So they're obligate anaerobes.
They are.
Which means they don't want any air.
They do not.
Facultative means they could go either way. Right?
Correct. These are not facultative. They have no mitochondrion. There's no way for them to process oxygen. And in fact, a lot of parasites that are obligate anaerobes have molecules in their cytoplasm which actually traps oxygen and gets rid of it and detoxifies it for them. Ascaris is one of those organisms. It's why it's a pink-colored worm. The myoglobin of the worm actually traps oxygen and keeps it out of the metabolism of the host. Oh, sure. Beautiful scanning electron micrograph picture of Giardia lamblia on Wikipedia.
We'll have to link to that.
Sure.
All right, so that's an example of
or we could also link to the book.
we will put a little badge on the website.
Giardia,
book.
By the way, there's a historic note to be made for Giardia also, although I haven't intended to talk about that mostly for this session, but this was the first protozoan ever viewed under the microscope.
Oh.
How do I know?
How do you know, Dick?
Because Leeuwenhoek described it.
His wee animalcules?
That's right.
Was one of them?
That is one of them.
So he got water from where?
No, not water. In this case, his own stool. No.
So he had this infection?
What was Leeuwenhoek doing looking at his own stool when there were so many other things to look at?
He was a little self-centered, I guess.
Perhaps.
Was he sick?
If you have Giardia, you're not well. Let's put it that way. You're not always
Dick,
sick.
Is everyone that has a Giardia infection ill? Or are they asymptomatic infections?
There are three kinds of people, actually. Three. Count them. 1, 2, and 3. The first kind is infected, asymptomatic. The second kind is infected, symptomatic. And the third kind is intermittently infected. In fact, I have a picture in my book of Leeuwenhoek looking at his own stool through his little microscope. By the way, which you can go see if you ever get to Leiden.
Right.
In Holland, you can actually see his microscope.
So the people who are infected without symptoms, can they be infected for years and shed it and infect others?
They can. They become carriers.
Carriers, yes.
That's right. So there are carriers for bacterial infections. I'm sure there are carriers for viral infections.
Absolutely.
And there are carriers for helminth infections.
Okay.
Even.
So Giardia is a flagellated protozoan parasite.
It has its own source of locomotion.
What family is it in? What's the higher?
Mastigophora.
Mastigophora.
That's right.
And the more generic name for it would be, it's a kind of what?
It's a flagellate.
It's just a flagellate.
Yep.
Is Trichomonas a flagellate?
It is, in fact. And
related to Giardia?
It is. Vince, you're asking all the right questions here, because it was flagell, the drug that was invented, or discovered, I should say, and developed to treat trichomoniasis. That was the original treatment.
I used to work on that.
Did you?
Between college and graduate school,
Really?
I worked for a year in a laboratory of a company that made flagell.
Yes.
And we used to assay every lot that was produced. We had trichomonads growing.
How interesting.
And we would add the flagell. We'd make dilutions and add them and time how long it would kill them under the scope.
Doing therapeutic indexes and stuff.
Yeah. So I used to work with that, and they always used to say, "Don't go to the bathroom after you're working with this." Okay.
Was it Trichomonas vaginalis?
It was vaginalis. Yes.
Really?
It was an ointment that contained flagell that was for female use, because it's a common female infection, right?
Extremely common.
Hence the name. Are there other trichomonads?
Tons of them.
Okay, so Giardia, so that's another non-vector.
There's one called Trichomonas foetus that causes abortion in cattle, for instance. So there's a whole slew of them. And in fact, most flagellated protozoans are free-living. So now you have to say some other thing about these groups. You have two kinds of organisms, right? You have those that are parasites and those that are not parasites. Do you know of any free-living viruses, Vince?
Free-living? Meaning they don't need a cell to reproduce?
Right. So it's impossible, right? The definition of a virus is that it's
an obligate intercellular parasite.
It's an obligate intercellular. But for protozoans and for helminths also, you have two different groups. You have groups that don't cause parasitism at all, that live free in the soil or water, and then you have the group that does.
So the Giardia could exist free-living? No.
No.
Needs beavers or something.
Well, the cyst form of this infection is long-lasting in the environment.
It's like a spore, basically.
It's like a spore.
Yeah, but that's not growing. It's not even alive by my definition.
It's not metabolizing. It's not metabolizing. You can't kill it with anti-metabolites.
Right.
Because it's not respiring even.
All right. So that's an example of a non-vector protozoan parasite.
Right. That's right.
Okay.
That's right.
What did you want to do next?
Well, let's talk about a vector-borne.
How about a vector-borne parasite?
Protozoan parasite. So, are there any flagellated vector-borne parasites?
Probably.
Well, you know one.
Vector-borne?
Give me
some hints.
I'll give you the biggest hint I can do.
Okay. Dick is snoring. Sleeping sickness.
Thank you, Vince.
Which is...
do we know the name of that organism?
Yeah, we do, but my 56-year-old brain is...
well, my 69-year-old brain has the answer.
Wait. Because, I know it's spread by the reduvid. Is that right? No.
That's the American form of trypanosomiasis. This is African trypanosomiasis.
Trypanosomes?
Yes.
I should know this because my wife used to work on them.
Well, that's the reason why
with George Cross.
Trying to give you a hint here. Transmitted by?
Tsetse fly.
Correct. Famous crossword puzzle.
TSE.
That's
TSE.
Right. That's right.
What's the global distribution of?
Just Africa. Sub-Saharan Africa.
Sub-Saharan Africa.
It's not even all over Africa.
So it's an old world parasite.
It's a very old world parasite.
It's not really elsewhere?
Nope.
Why is that?
That's a hell of a question, isn't it?
Well, you asked the same for viruses.
No, and I'll tell you the answer relates to the fact that it's vector-borne.
The tsetse fly only in Africa?
Yeah.
Okay.
You can't find it anywhere else.
How big is a tsetse fly?
It's as big as a housefly.
Okay. And they bite you and they deliver this?
They're a biting fly. This is correct. And they're found both in East and West Africa. And there are actually two varieties of African trypanosomes. One of them is called Trypanosoma gambiense. And the other is called Trypanosoma rhodesiense. Now, where is Rhodesia?
Southern Africa.
No. It isn't in existence anymore. It's now called Zimbabwe.
It used to be, right.
So we should call this Trypanosoma zimbabweense.
But we're not going to change it.
We're not going to change the name.
And gambiense refers to?
West Africa.
Gambia.
Yeah.
I'd recall that word from mosquito species, perhaps?
Absolutely. Anopheles gambiae.
Okay.
So, but it's now Senegambia.
Okay.
So, because Senegal and Gambia have
protozoan,
sort of merged.
Giardia and trypanosomes.
That's right.
And trypanosomes cause sleeping sickness.
They can.
They can, not always.
Not always.
Is this a big deal?
It's a huge deal because it not only causes it in people, it causes it in European cattle.
Which are brought to Africa to grow?
Apparently. So there isn't a native cattle called the Ndama cattle in West Africa. Very, very difficult to raise. The meat is very tough and the yield is very low. But they are resistant to West African trypanosomiasis. On the other hand, every game animal in East Africa has the organism.
And they're fine.
And they're fine.
So you have to cook your game.
That's the least of your worries. You have to worry about being bitten by a tsetse fly.
I see. So these animals are fine. Why is that?
That raises the $64 billion question, because the real name for the organism is Trypanosoma brucei. And who was Bruce?
Don't know.
He was the discoverer of brucellosis.
Mm.
So while working on the causative...
It's a bacterial disease.
It's a bacterial disease that causes abortion in cattle.
Right.
Brucella abortus is the actual name of this infection. It's also called shipper's disease in cattle terms, because the cattle, when they're being shipped, if they've got brucellosis, they'll die from it. All right, so if you go to Africa and you trace this guy's history, he started out on the island of Malta, a Brit, right, that goes to the old world to discover yet another parasite. And that there was a stream of British parasitologists that spread out all over the world. Sir Patrick Manson was one of those people. The guy who got the Nobel Prize for working out the fact that mosquitoes transmit malaria, whose name is... Oh, come on, you know this guy.
Walter Reed?
He was an American.
He did yellow fever anyway.
Yeah,
right?
I mean, who was given credit for discovering the transmission cycle for malaria?
I don't know.
You don't know? Well...
Give me a hint.
I will, as soon as I look it up.
Oh, that's why you don't know. So this is malaria?
Malaria. Now, this is a quiz for the listening audience, because I actually do know the answer to this question. But I'm just holding back now, hoping that you will find the answer.
There he is.
Before.
Is this guy here, Charles Alphonse Laveran?
No, Laveran was one of the discoverers.
Camillo Golgi?
No. Keep going.
I didn't know that he was involved in malaria.
No,
He's the guy who discovered the Golgi apparatus.
That's right. Who discovered?
Ronald Ross?
There you go.
Really?
Sir Ronald Ross.
A British officer in the Indian Medical Service.
That's right, and he discovered that canary malaria was transmitted by the Culex mosquito.
And he did this in 1897
He did.
and got the Nobel Prize in 1902.
That's right, along with Laveran and several other people.
Must have been one of the earliest Nobels, right?
It was one of the earliest Nobels. But he did not discover the mosquito that transmits malaria to people. That was discovered by Grassi in Rome, Italy.
Now, how did we get into malaria here?
I don't know. Vector-borne mosquitoes, Africa.
Is it a protozoan?
We're going. Yes, it is. But we're drifting. We are drifting.
Trypanosomes was
We are
an example.
drifting.
Malaria is another example.
Wait, I wanted to tell you why.
Vector-borne.
You asked me the question before as to why all the game animals in East Africa get infected, but
they're fine.
But
right?
they don't get disease.
Yeah.
And this was the crux of Bruce's work, to look at the difference between East African trypanosomiasis and West African trypanosomiasis, and ask the question, why, when an animal encounters West African trypanosomiasis, they die? And when they encounter East African trypanosomiasis, they live. And he, for years and years and years, he could not discern the reason. He tried everything in his power to find out, but he didn't have the tools to investigate as to what was going on here. So he worked on this for 20 years and then just gave it up. Okay, he just quit. He said, that's enough. I've done enough work here. I can't take it any further. I'm out of here. And it remained an enigma until very recent times, when Dr. Mickey Rifkin and other people discovered that there is a susceptibility to the West African trypanosomes to HDLs, as I recall. I'll get the story straight. It's not time to talk about this yet.
Okay.
But there's a lipoprotein in the blood which limits the infection in animals.
Okay. So this is, can I ask?
You may.
So the Giardia you ingest.
You do.
They replicate in your intestine.
Oh, we get to routes of transmission next. So
no, the trypanosomes are injected in your blood, though, right?
They
you
are.
don't ingest those.
No, you don't.
If you ingest them, they won't do anything, I
They
presume.
don't.
Okay. So some parasites are in your gut, some are in your blood, or some in your respiratory tract. Protozoan parasites.
A protozoan parasite in your respiratory tract. There's one called Pneumocystis carinii. It's thought at one time it was a protozoan, but it actually turns out, based on its 16 sRNA, to actually be a fungus.
Okay. All right. Go ahead.
Go ahead, he says.
Now, you have protozoans that are vector and non-vector.
That's right. And we have
helminths
that are vector-borne and non-vector-borne. And I'll give you an example of a helminth that's vector-borne, okay? It's called filariasis. So filariasis actually turns out to be a general term for a group of worms which are transmitted by the bites of mosquitoes. That's interesting, right?
How can a worm fit through a mosquito thing?
Exactly. When you look at the size of the worm that's actually taken up by the mosquito, it's quite small. and it's in circulating blood. So we have several groups of helminths that fall into this category of filariasis. One of them actually turns out to be a parasite which is not transmitted by mosquitoes, but rather it's transmitted by black flies.
Mm-hmm.
And it's called river blindness, Onchocerca volvulus. Now, I know that you said your wife works at Merck.
Yes.
They were the discoverers of the drug, which is actually very effective against filariasis. And you know the name of that drug? Go ahead, Vince, tell us.
Ivermectin.
Yes, ivermectin. And do you know the story of its discovery?
No.
I do.
But I heard the name for many years.
Do you even know who discovered it?
I do not.
Well, a very dear friend of mine, William Campbell, discovered
Bill Campbell. He used to be at Merck.
He did. He was at Merck. He's now at Drew University.
I think he gave us a snake many years ago. He used to breed them.
Really?
Does that sound reasonable?
Snakes?
No.
I never knew that about him, but he was a very dear friend of mine. So Bill Campbell actually was at Merck when the executive returned from Japan with a divot from a Japanese golf course. And this divot was then sent to the laboratory and placed into a number of different culture media. And in one of those broths that they got, which they then shipped out to various laboratories for testing, turned out to be an extremely effective drug against a particular parasite, namely, in this case, dog heartworm. Okay, now
Which is a helminth?
It's a helminth. It's a vector-borne filarial parasite.
What's the vector?
Mosquitoes. The Culex pipiens mosquito transmits it. Okay?
So this is what you give ivermectin to dogs. It's called HeartGuard, I think.
Correct.
Because they get a heartworm transmitted by a mosquito?
They do.
Really?
They do. And the worm actually lives in the ventricle of the heart.
Causes damage, I presume.
You'd think so, wouldn't you? But you know what its main effect is? It cuts down on the rate of respiration by slowing down the flow of blood to the lungs. And the dog starts to respire at a lower rate. Its metabolic rate drops. It becomes susceptible to pneumonia, and then it dies from that.
Okay. So that's why you give it HeartGuard.
That's correct.
Okay. And so this is an example of a filariasis.
Which is vector-borne. All of the filaria are vector-borne. All of them.
And what was the causative agent of river blindness again?
It's called Onchocerca volvulus.
Okay.
Onchocerca volvulus. We'll come back to all of this
I'm sure we will.
later, but I'm giving you the breadth and the depth of my
I'm
field.
fascinated. That's why I'm asking questions.
It's fine, Vince. It's fine. You're allowed. In fact, that's how this show will proceed. We'll do it Socratically.
Ivermectin. And then Merck gave it away.
They are still
to
giving it
Africa
away. They're still
for
doing
river blindness. So it was originally picked up for dogs and then found to work on river blindness? Is that right?
The screen was using a dog heartworm assay. They had dogs infected with heartworm. They would take culture broths from various places, or various things, I should say, various soil samples primarily, okay? They'd let things grow out in it. They would divide up the broth and ship it off to the labs, and they would test it for antimicrobial activities, as well as other activities, too. That's how they used to screen. I think it's all done by computer now. That was a bit of a joke, because Bill Campbell was never a big advocate of that. He was always of the mind that the drug industry is one step away from the next magic bullet discovery.
Yes.
And I think it's been that way for a long time. So it's hard to predict when the next silver bullet will be found, but indeed. Okay.
So I just found a paper here. Cloning and characterization of the ivermectin receptor in filarial nematodes.
And who did that work?
Well, that's my wife. She was involved in that. She went to Merck. One of the things was to find the receptor for ivermectin.
Wasn't that gamma amino benzoic acid?
No, the receptor, which was a channel.
Chloride ion channel. That's right.
It's an ion channel.
Wasn't that with vasculitis? It was with...
the Greek fella.
That's right.
I can't remember his name.
I'm
Dimitri
vasculitis.
So they did it in C. elegans.
Right.
Because they are also sensitive to the drug.
That is exactly right.
And it has good genetics, so they were able to do that.
Yep.
But C. elegans is a flatworm?
No, it's a roundworm, and it lives in soil, and it's free living.
But it's not a filarial. It's not a helminth.
It is a helminth.
Well, that's why you would be susceptible, I suppose.
Sure. Helminths in general have similar biologies.
Okay. Sorry for the diversion.
Not at all. So here's an example of a vector-borne helminth. Now, what about a non-vector-borne helminth? Well, I'll give you the best example I know is Ascoras lumbricoides. Ascoras Lumbricoides. Now,
Yep. This the one from snails?
no. No,
It's
no,
a worm.
stop.
It's a worm.
It's a worm the size of the pen you just wrote its name in.
And it goes in your intestine,
It
right?
does. It does. And you'll never guess who named this worm.
Mr. Lumbricoid? Sorry.
Linnaeus.
Linnaeus did?
Yes. You know
Okay.
why he named it Lumbricoides?
No.
Do you know what the name of the earthworm is?
So Annelida something or
No,
other.
it's Lumbricus terrestrius.
Annelida is the family, I think, right?
So, stop. You can stop right there, Vince. There are 7 different ways for a virus to replicate, and they all have to end up with messenger RNA.
Yeah, you remember that.
I do.
And I don't remember
it's
this.
the Baltimore scheme.
All right, I'll get this. You have to give me. You have to teach me first.
Here's the book. Go home and read it. So Linnaeus.
I don't have time. I have to prepare for TWiP.
And so do I, by the way. I have to think up some stupid questions so you can give up some
I think
smart
they
answers.
just come automatically, Dick.
So Ascaris lumbricoides reminded Linnaeus of the earthworm in its size, but there's a basic difference. And that is the earthworm is segmented, and Ascaris is not segmented. In fact, no nematodes are segmented. They're unsegmented roundworms.
What's the significance of a segment?
Well, it has something to do with its embryology.
Does it provide any advantage?
I guess if you want a multiple set of brains, yeah, sure.
There's a brain in each segment?
There can be.
Mm.
There isn't always, but there can be.
So maybe the segments were free living at one time.
Well, Ascaris' nervous system is the best studied invertebrate nervous system in the world because you can see it. The worm is the size of this pen. You can actually pin this worm open, and you can show all of the commissures and the lateral branches to the nervous system. There was a guy by the name of Tony Stretton that worked at the University of Wisconsin, as I recall, who actually did a physical map of Ascaris. Yeah, it's big. He's got
not
a picture.
Segmented.
It's not, of course not.
Shiny.
It's pink in color.
How long can they get? 40 centimeters.
Big.
Half a meter.
They're big.
Wow.
Yeah, and there are many varieties of Ascaris. So Ascaris lumbricoides is the human infecting Ascaris, and Ascaris sum, S-U-U-M, infects pigs, and they're essentially the same worm. They're indistinguishable by morphology. You'd have to do the genetics to make sure of the differences.
Dick, it says here 1.2 billion people are infected with these.
That's a minimum.
Ascaris.
That's a minimum estimate.
So it's a global thing.
Do
we have it in the U.S.?
Of course we do. It's all throughout Appalachia, throughout the lower 48. I would say the Gulf Coast states are loaded with it.
Does it kill you?
It can, but it usually doesn't.
And there are treatments, right?
There is the treatment, of course, and that's called mebendazole. Mebendazole. You'll never guess who makes it. You're right, Merck makes it, and it's all derived from the benzimidazoles, thiabendazole being the parent drug. So that was invented quite a ways ago.
The filaria, are they also global in distribution?
No, they're not. They're restricted by their insect
Vectors. But because Ascaris is a non-vector,
right.
It can be
And
everywhere.
there's one other feature of this worm that you'll be impressed with. It produces 200,000 eggs every day.
Wow.
And it can live for up to 5 years. So if you do the math, that's a prodigious amount of eggs. Now that means its reproductive strategy is it will spread itself throughout the entire world and hope for the best. And it works.
How's it transmitted? I guess we'll go into that, right?
No, I can tell you right now. This worm lives in your small intestinal tract. It sheds the eggs into the lumen, and the eggs eventually become incorporated into the feces, which is then passed out. So it means that it's a fecal-transmitted oral fecal route. That's how you catch it.
It's related to hookworm?
It's a worm, yeah, and it's non-segmented, and it's a helminth. That's about as close as you get.
It's non-vector
It's non-vector transmitted.
Okay.
But hookworm and Ascaris are pretty far apart on the phylogenetic tree of life in terms of helminths. Okay, but there's a lot of organisms out there that have Ascarises all to themselves. There's Toxicara, which is not exactly Ascaris, but it infects dogs and cats, so you have Toxicara canis and Toxicara cati. And both of those Toxicara compared to the Ascaris worm. If you had an immature human Ascaris lumbricoides next to a mature Toxocara canis, you couldn't tell them apart. They both basically look alike. The eggs are quite different.
Okay.
So there are lots of differences here, but the general pattern, they would call this, because it's transmitted from feces from person to person, but because the feces has to be in the soil to begin with, they're called geohelminths. So there's a whole family of worms not related to each other, but whose eggs are found in feces, and the feces contaminates the soil, and the soil contaminates your environment, and therefore you get the infection. So those are called geohelminths. And trichiura, the whipworm, is in that category. Hookworm is in that category. Strongyloides is in that category. Ascaris, and a few others.
So it sounds like Ascaris is something medically important that physicians in this country would see often.
They see it quite a bit in our clinics because, of course, we're on the East Coast and we got a lot of immigrant populations coming to New York from places in the Caribbean and South America and Africa. So we do see a lot of it, yeah. In fact, Vince, I think I might have mentioned this to you in our very first TWiP.
TWiP?
No, TWiP. In our first TWiP, I began my career here as a technician.
Yeah, you mentioned that in the first TWiP.
And also in the first TWiP.
And you just told the story of how you went to work with
that's
Dr.
right.
Brown. And that was, you'd seen some Ascaris in that.
Yeah. Right? Yep. Well, we saw that every day. In fact, this was during the height of the immigration from Puerto Rico.
So that was the major parasite you saw?
We saw 3,
yes,
classically, and they were called the unholy trinity. One is Ascaris, one is Trichuris, and the other is hookworm.
What's Trichuris?
Trichuris trichiura is another parasite that lives in the gut tract. In this case,
worm?
It's a worm. Yeah, it's a helminth.
Okay.
It produces a long-lived egg in the environment, and Ascaris eggs can live up to 10 years in the environment, depending on the soil types and the temperature profiles. So there's an ecology to all of this also. Just like there is with viruses, just like there's with bacteria and fungi, protozoans, helminths, there's a huge ecology associated with this. And in fact, because the feces is deposited in soil, the feet of birds and earthworms actually help to redistribute the eggs from the fecal mass to various spreading out zones from the original deposition zone. So a lot of ecology goes on here.
Dick, I think it's beginning to sound to me that these parasites are more distributed than I ever appreciated.
Well, if you ask how many people in the world are infected with at least one of the parasites that we're going to be discussing here, namely either protozoans or helminths that are distributed by fecal contamination, I would estimate that half of the world is infected with one of them.
Wow.
And many are infected with multiple
probably more
infections.
than are infected with viruses. What do you think? Are there parasites, other protozoans or helminths in the oceans?
There are.
I know there's some in water.
There are. In fact, we'll do a whole show on sushi. All right?
Excellent.
And the reason why we're going to do it is to give you a sense of history as to how far we've come with regards to what we know now versus what we didn't know when we first discovered what was going on with regards to the transmission of a specific parasite which infects cetaceans.
Whales.
And dolphins.
Mammals.
Yeah.
Mammals of the sea.
Sea mammals. And the way they catch their infection, of course, is to eat fish. And the fish are infected, right? So the way we catch this aberrant parasite now, because we don't look like cetaceans to the parasite, is we eat those same fish.
So I shouldn't eat sushi?
I didn't say that.
All right. Well, we'll wait for the sushi episode.
That's right, but I'll keep the audience at the edge of their chairs.
We have to name these episodes, you know.
We do.
We didn't name this one yet.
No, this is...
But you can help me with that.
General Parasitism, Part 1.
What was last week's?
Introduction.
Last month, sorry.
The introduction to TWiP.
Intro to TWiP.
So here we have general characteristics, and we're just going through a bunch of categories of the way these parasites find their way into their hosts. So they find their way in by fecal contamination of food and water, by being delivered to your doorstep by a vector, okay, and finally by sexual transmission.
What are some of those?
Well, I can only think of one, to be honest with you.
Cryptococcus neosporans.
No, that's a fungal infection. Stop it. You can't fool me. Oh, yes, you can. Actually, you can fool me quite easily.
No,
this is Trichomonas vaginalis. Trichomonas vaginalis is a sexually transmitted infection.
Is it symptomatic in both sexes?
No, in fact, it's not symptomatic in males at all. And that's the reason why it's so easily transmitted from person to person.
That's the only one you can think of.
It's the only one I can think of.
Do you teach that in your course?
If you are a homosexual,
yes,
you can also transmit Entamoeba histolytica sexually if you'll consider anal intercourse as a sexual activity, because then the cyst of the amoeba can be picked up on the penis of the partner that inserted himself into.
And what do they do with it then?
Well, then they
can transfer it to someone else
via fellatio
or intercourse of other. No,
no,
because this is an intestinal.
It has
parasite.
to pass through the intestinal tract.
I see.
Yep. So there is that.
So Entamoeba is a protozoan.
It is. It's a non-vector-borne protozoan. In fact, it's a geo. You'd call that a geoprotozoan.
But we'll learn about this, right?
We will. We'll go through each one of these, in fact. We've got lots of episodes to go through.
Wait.
Oh, we're going to have so much fun here. And I'm finally talking about something I was trained in.
You speak very well when you talk about something you're trained in. Not that you don't speak well on TWiP.
Oh, well, thank you, Vince.
But here, your train of thought is unimpeded. Your cadence is excellent. There's no hesitation because this is your stuff.
You are right.
Are you the world's expert on parasites? Eukaryotic parasites? Who is?
I don't think there's any one person that can know everything about all of these.
But you are an expert.
It helps to have written a book on it, and we're in our 5th edition. So every time we revise the book, you relearn everything.
Yes.
And I don't write the helminth section, although I did all my research on worms.
Yours was a Trichinella thing, right?
Yes, and I'll devote a whole issue to that.
And that's a worm?
Oh, it's a nematode.
Segmented?
None. All the infectious nematodes are non-segmented.
Non-vector borne.
No, some are vector-borne. We've already covered that.
No, the Trichinella.
Trichinella is not vector-borne, but you catch it by eating raw or undercooked meat, and so that means that there's another way of catching these parasites.
By eating. We said oral fecal contamination, but that's different because
that's
meat.
Different.
So why didn't you bring up the Trichinella in this list here? I'm surprised.
It's going to come in. Foodborne infections. So there are foodborne infections.
We had vector and non-vector. Non-vector is foodborne, right?
That's right. But in this case, the stage of this parasite is encysted in muscle tissue. There are some protozoans that do this as well. There's one called Sarcocystis, which is actually part of the Eimeria group that infects the same way.
But this Trichinella insists itself?
It does.
Would that be the picture on our logo?
It actually is.
And what's that called?
That's called the nurse cell.
Why is that?
Well, because what you're looking at on that picture is a coiled up worm inside of host tissue. That host tissue used to be striated skeletal muscle cell. Imagine what that looked like as a striated skeletal muscle cell, and now compare that to the nurse cell, and you will see that there's no similarity whatsoever.
Why do they call it a nurse cell?
Why did I call it the nurse cell?
You named it?
I did.
So if you go to Wikipedia and you look up nurse cell?
I don't know. Let's see if it's there. That'll be a good test of Wikipedia. If it's not, we can add it.
Well, Dick, you need to have an entry. Yeah, there is a small entry.
What does it say?
Human physiology, microbiology. Let's see. Is a term to describe an infected cell in trichinosis.
That's the one.
It doesn't give you any credit, though.
Why should it?
I'll have to put an entry for you.
I'm not...
Let's see if there is one. Despommier. Dickson, Depomier, you do have an entry.
Of what?
Oh, you've got many. You have vertical farming. You have... Look at this. You've got a brief one here. And then you have one on vertical farming. So there is a vertical farming entry.
I'm sure there's one for parasites too, though.
And you're in here.
Oh, okay.
Cool. So then you have a small page on you. Then they have toxocariasis.
Right.
And your name is in there.
Because I wrote a review.
They have trichinosis, and your name is in there as well.
Yeah.
But the entry on you is one paragraph.
Maybe that's all I deserve.
It says you're a professor of public health, so we'll have to amend that.
We will.
He conducts research on intracellular parasitism.
Yep.
Teaches courses on parasitic diseases, medical ecology, and ecology. In recent years, Depomier has received considerable media coverage for his ideas on vertical farming. That's true. You're a famous guy.
Oh, absolutely.
So, trichinella, we'll talk about that as well.
Sure.
All right. I'm sorry to have diverted you again.
That's okay, Jens.
That's what happens in TWiP.
It does, and TWiP also.
But, we never said in TWiP what the icon in TWiP is. We'll have to explain that on a TWiP episode.
That's true.
Now we know it's a nurse cell.
That's true.
In
So
TWiP.
a lot of parasites, a lot of eukaryotic parasites alter their environment, especially the intracellular ones.
Mm-hmm.
And now there's another set of categories of parasites. And I want you to write this down too now, because this is very important.
All right.
So far we have vector-borne and non-vector-borne.
Right.
And then we have now we have another category of parasites. And we have long-lived parasites and short-lived parasites.
So that's going to overlap with the vector and non-vector for sure.
It will shake out in a totally different direction. Now, what I'm going to suggest to you is quite, how should I say this? It's speculative, and yet associated with the speculation is proof of concept, and I'll give you one proof of concept. Long-lived parasites. When I'm talking long-lived now, Vince, I'm talking 10 to 20 to 30 years of life in the same host. All right, you mentioned one that you can catch by waiting in a rice paddy. Schistosomes.
Mm-hmm.
The adults of those parasites live in your bloodstream. In the bloodstream. In the face of the height of the immune response. They're inundated with antibodies and all kinds of reactive cell types. And yet they live there for 25 years. Now, how do they do that?
Good question.
We're going to talk about this.
I'm sure.
The point is, if we could find out the exact mechanism of how they do this, we might have a medical intervention strategy for allograft and homografts. We might be able to transplant your tissue into me or my tissue into you.
Well, we can do it, but we suppress
the heterograft response is totally diminished by looking at the biology of schistosomes and figuring out how such a large worm that you can see with your naked eye avoids immune attack from the host. Over 25 years.
Schistosoma japonicum.
That's the one. Well, there are two others. Actually,
Mansoni.
Wow, listen to him.
What else?
This man you know much more than I thought.
I just heard you talk over the years.
Well, there's Schistosoma hematobium, Schistosoma mansoni, Schistosoma japonicum, and a fourth species has been recently discovered in Vietnam along the Mekong Delta, and it's called Schistosoma mekongi. It's related to japonicum, but it's not same.
Do we have any in this country, in the U.S.? Zero? Europe?
We don't have any human schistosomes in this country, but we do have bird schistosomes. They're a different genera. They're called Schistosomaceae.
And in Europe, are there?
Nope.
Africa?
Oh, yes.
What does it relate to? There's not vector.
Snails.
Snail is the vector.
Well, if you think about vectors, then vectors bite you and introduce something. Snails don't actually bite you.
They don't have to bite, do they? A vector just transmits it, doesn't it?
No. A vector actually bites.
Really?
A vector bites. So a snail is called an intermediate host.
Mm-hmm.
All right? It harbors certain stages of the parasite which must develop within the snail before emerging from the snail and seeking out a mammal.
Okay.
So that's quite different from a vector. Snails are not vectors. Snails are intermediate hosts. I can prove it to you.
Is there a vector?
For what?
Schistosomes.
No.
It goes from snail to snail?
No, it goes from human to snail to human to snail to human
Is
to
human
snail.
required part of the cycle?
It is.
Without humans, there would be no schistosomes?
Except for Schistosoma japonicum, in which case there is the ox, or the water buffalo rather,
right,
and monkeys. In Africa, there are a few reservoir hosts. Those are called reservoir hosts. That is another animal species that can harbor the same stages that we can. That's the definition of a reservoir host. So then we have to break out what is a vector, what is an intermediate host, what is a definitive host, and what is a reservoir host. We have all these terms to deal with in terms of parasitic infections because we have all these permutations, and they took years and years and years to discern. The people who were responsible for these are fascinating people of history, and they altered the landscape for medicine as the result of their pioneering work. People like Sir Patrick Manson and Ronald Ross and Laveran and those kinds of people. Wutcher and Bancroft. Put them together, and what do you got? Wuchereria bancrofti, the one that causes elephantiasis.
It's a parasite?
It's a parasite. It's a filarial parasite that's mosquito-borne. It causes elephantiasis in people.
Right.
So we have all these colorful, wonderful things to talk about. There's another one called Loa. Easy to remember. It's transmitted by a tabanid fly. They're related to horseflies, deer flies.
So do you have any short-lived or many short-lived parasites?
Lots of them. Okay, but what I want to say about the
long-lived?
We're not interested in the biology of short-lived parasites. You know why? Because our immune system kicks them out and we're done with them.
What does the short refer to? How long? Less than a year?
Depends. You know, like, say, for instance, Giardia. In most normal people, you'll get infected with Giardia. If you don't get treated, it might last a month, it might last 2 months, and then it's gone.
But they don't go in your bloodstream, right?
Nope.
They stay in your intestine.
Yep.
So what eliminates a Giardia infection from your intestine?
Good question, Vince. What would you suppose?
Well, I know there are antibodies in your intestine.
Which kind?
IgA.
There you go.
Secretory IgA.
There you go.
Is
so
that
the same kind of antibodies that would eliminate a viral infection at the mucous surface would also be important to interfacing with the Giardia.
Does complement play a role? These are membrane-bound parasites. No.
Complement is a blood protein, set of blood proteins, correct?
Yes, it's not in the lumen at all.
So it couldn't possibly get to Giardia.
How about a cellular immune response?
You might see at the surface of the attachment to the columnar cell some accumulation of reactive cells, but mostly no.
Can I have one of these?
I brought them just for you. You might want to tell the audience. Now we're going to have a word from...
We don't have a sponsor for this show yet.
Yes, we do. Now we do.
We have Dick brought some chocolates with him.
He did.
Fine hazelnut chocolates, Ferrero Rocher.
Which is one of my favorites.
I am hungry. And would you like one, Dick?
No, I've had mine already. Thank you.
So the short-lived are not as interesting because they cannot evade immune responses.
That's correct.
That's an interesting perspective, but I'm sure people do work on them, right?
Oh, yes. Because they still are medically important. And they can be fought off with some wonderful drugs that have been developed for that purpose. But it's the long-lived ones that I want to emphasize here because of their biology. The biology of a long-lived parasite suggests strongly that it has evolved ways of evading the immune response, or subverting the immune response, or hiding out in a place in the body that has no immune response associated with it, such as the anterior chamber of your eye, or the cerebrospinal fluid, for instance. So parasites that live a long time are of medical interest because if you could discern their secrets, you could develop a science called parapharmacology, in which we're making use of substances derived from parasites to augment our pharmacopoeia of drugs, which we're running out of, by the way, for diseases such as diabetes type 2. When I describe the biology of Trichinella, you'll see what I mean, because the worm has the capacity for inducing an angiogenic response of a specific kind, a very specific kind. In fact, no other parasite induces that kind. Maybe Onchocerca would join the group, and the group of 2 that can do this, but none others. So if we could find out how it does it, we might be able to apply that to some medical solutions.
You know, there's a similar story with viruses that persist in their hosts for long periods of time.
Sure.
Often lifetimes.
Sure.
They either have to go to a place where they're not detected.
Yep.
Or they have to actively antagonize the immune response.
Or they remain latent.
Or they can remain latent.
Yep.
And understanding, that's why we study those viruses, because understanding how they do that can give us therapeutic approaches.
Correct.
Right?
Correct.
Cool.
So the problem with my having just said that is that there is very little governmental support at the NIH supporting level for the biology of some of these parasites.
Why is that?
I think because they are low profile in terms of medical importance. They've been perceived as sort of background noise.
How is parasitic research funded compared to virology? Less?
I don't know what the numbers are, but I know that it's going to be less because HIV falls into the virus category, and that's huge amounts of money for that. Whereas malaria garners the the lion's share of NIH money for parasites. I used to serve on a study section that gave away a lot of money, but never as much as we needed, and certainly not enough to cover the fundamental work on the biology of these organisms. For instance, we do not know what Trichiura eats. We don't even know what it eats. How could you not know something about that, about an organism that you can see with your naked eye? We could look at this organism all day long.
Is that a worm?
It's a worm. You'd never figure out what it eats by looking at it. We don't know what Trichinella eats.
Maybe it eats Ferrero Rocher.
We do know what Ascoras eats.
You have to feed them things and see what they will eat, right?
Well, that means you have to work with them, doesn't it? And to get that money means that you have to justify it. And that's the difficult part.
Can you do genetics of parasites? Malaria, I guess you can.
Sure. You can transfect these organisms. Cenoibritis elegans, of course you can.
Trypanosomes.
Of course you can. Of course.
Fascinating.
Yep. They are fascinating. So I hope over the next, 2 to 5 years of TWiP that we'll find some time for some specific stories that relate to vector-borne protozoans, vector-borne helminths, long-lived parasites, and their stories, because I think their stories are worth broadcasting and getting people interested in. It's held my attention for more than 38 years. I began my graduate days in 1964. So since 1964, I have maintained, without any help, the parasites themselves have presented to me so many interesting biologically based problems that it has captured my attention and kept it for those many years. So I'm grateful to Vince for the opportunity of presenting them once again to a larger audience through the iPodcast that we've developed. And for that, I want to end this session by thanking you, Vince, for...
I understand why you work on parasites. I'm beginning to understand. It's very interesting.
Well,
and you're going to reach more people on this cast than you have in your whole career of teaching students.
I think that's a remarkable
How many
achievement.
students have you lectured to in your whole career? Would you just guess?
From 1971 to the present, every medical school class consists of about 150 medical students. So let's make it 38 years. 38 times 150 is how much? You have a calculator?
Yeah, no, I have a brain.
Has a brain. That's even better.
38 times 150.
Come on, he's doing the math as we
40,
speak.
12.
Here we go.
5. That would be...
Big number.
About 7,000.
7,000 medical students. And you could add about half as much again for school of public health students.
Within a year, you'll have surpassed
So
that.
make it 10,000 students
Yeah, you
altogether.
can reach more people.
And I used to teach at NYU and at Cornell too sometimes. So
In a month, you could outdo that. The point is that you can take what you have restricted to teaching to students at this institution and now instruct the world, whoever wants to listen. I think that's powerful.
I do too.
So you need to do a good job.
So I better go home and read my book.
I don't think you need to. What are we talking about next time?
Next time, I think we should pick a specific story and start with that story.
All right, that'll be great.
So I think malaria would be the appropriate topic.
Now, as of this recording, which will eventually go on the website of ASM, American Society for Microbiology, which is called microbeworld.org, and I believe the website will be microbeworld.org/TWiP.
Right.
And there'll be an access email there to send us questions, because we do want listener questions, comments, requests. It's my phone.
There's one now.
So microbeworld.org/TWiP, and there'll be an access there for email, and you can send us your questions. And we thank everyone for listening. Tell your friends about us. If we're not already on iTunes by now, we should be, so you'll be able to find us there or at microbeworld.org/TWiP. You've been listening to This Week in Parasitism, a podcast all about eukaryotic parasites. Thanks for listening, everyone. Thank you, Dick.
Hey, Vince, it's a pleasure as usual.
I enjoyed it a lot. See everyone next month. That's December. And
Christmas.
for the presents. Another TWiP is parasitic.